Okay. Hi everyone. I'm Sophie, and my group with Maggie and Claire, we worked on the Machine Learning Challenge A for our project. And the main question we were trying to address is, are we able to predict how a cell will respond to stress in its environment? In answering this question, we created a machine learning algorithm that attempted to take a cell's expression line and be able to predict how it will affect to a certain environmental stress. And we also evaluated that learning algorithm and determined future work for our project. So our project used gene ontology analysis, and it was performed for a specific set of cell lines in our project. Each cell line was either negatively or positively correlated for a go term, which the go term is what tells us which bodily function is associated with that cell line. To produce our machine learning algorithm, we made use of two different data sets, one that had gene expression profiles and their related Go annotations. Not all of the cell lines had Go annotations, and that is what we split into our training and test data sets. And then we used the k-nearest neighbors algorithm, and we used a k value of 6 because numbers below that value had less predictions made, and numbers higher than 6 didn't change significantly in the number of predictions made, if at all. The cell line data set had some double mutant outliers in it that made those cell lines appear either extra fit or unfit. And so we kind of played around with different data pre-processing techniques and ended up testing out the regular raw data compared to a normalized version of the data, and then also compared to a data set where we got rid of the outliers and then normalized the data. We then submitted our predictions to Kaggle to see how well they performed, and we worked on evaluating the differences between the different pre-processing steps that we chose.
Our Kaggle scores were accurate for predicting Go terms about 52% of the time for the raw data, about 51% of the time for the normalized data, and just under 50% of the time for the normalized data with outliers removed. This was surprising to us as we thought normalization and the removal of outliers would lead to more accurate prediction. However, these scores are similar to others where scores range from 51 to 59%.
To assess the differences in the Go term enrichment between the data sets, we plotted the 5 most highly enriched terms for each of the types of data sets. So we have the raw and normalized enriched terms here, which had 2 of the terms in common, but they were all at different levels. And then we also have the normalized without the outliers data set that had 1 shared term with the raw and the normalized data sets. The term 0006952 was enriched in all data sets, and it's associated with the body's defense response to foreign bodies or to disease. And then term 0006874 was enriched in both the raw and normalized data sets, and it is associated with the homeostasis of calcium ions, which is necessary for almost all bodily functions.
In conclusion, our raw data set with the k-nearest neighbors algorithm is currently predicting the most accurate at 52%. This is on the lower end of the Kaggle submissions, and we're interested to see what other groups working on this challenge have done to mitigate the issues. Further directions for our algorithm include assessing if the K value needs to change depending on data preprocessing, trying different unsupervised machine learning algorithm techniques like hierarchical clustering, principal component analysis, or an association rule algorithm. We also think it's still wise to control for the double mutant data types, and clearly getting rid of outliers didn't work. So we would suggest splitting the data set into a normal set and a double mutant set, training the machine learning algorithm to recognize if it's a double mutant or not, and evaluating based on that criteria.